Welcome everyone to today's GlycoNet joint webinar with the ACS Carbohydrate Division. And it is my pleasure to introduce our speakers today. And our first speaker is truly a card-carrying glycoscientist, Professor Samy Cecioni from the University of Montreal. Samy is currently an assistant professor at the Department of Chemistry at the University of Montreal, and he completed his PhD in France, dividing his time between glycochemistry and glycobiology, so glycochemistry in Lyon and glycobiology in Grenoble. He then joined the lab of Professor David Vocatolo at Simon Fraser to work on chemical glycobiology, geared toward eukaryotic protein glycosylation. He was a CIHR postdoctoral fellow while he was working with David. And Samy is a nice example of progression through the GlycoNet system in that he was a trainee and is now a faculty member.
Thank you again, Warren, for the invitation and those kind words. I'm truly grateful today because, I mean, first for the invitation, obviously, but also grateful to GlycoNet because, as you said, I used to be a GlycoNet HQP in the lab of David Vocatolo, and now I'm a assistant prophet in Montreal. And GlycoNet support has played a large part in that transition and in this work as well. So today I'd like to talk to you about the main project actually that enabled me to do that transition. And once again, that was generously supported by GlycoNet. And I'll give a couple of examples of projects that were specifically supported by GlycoNet. But in general, the work I'm going to be discussing deals with chemical strategies that enable live cell quantitative imaging of glycosidase activity. So this is certainly not an audience that needs to be reminded how important the landscape of glycosylated molecule is for human health and disease. But I always like to state that this is all possible. You know, the tremendous diversity of glycosylated structure is all possible thanks to a finely tuned and regulated glycan synthesis and glycan recycling machinery. And this is particularly relevant to GlycoNet's mission, I think, because of the numerous diseases that arise from defect in glycosylation synthesis or glycosylation degradation. So the work I'm going to be talking about today really deals with that process of glycan degradation, in particular what happens within the lysosomes, that are these organelles that play a large part in recycling glycans. So inside lysosome, this would be an example that you may be familiar with of such a degradation pathway. And basically, you have glycolipids, complex glycolipids that are degraded one sugar at a time by exoacting glycosidases. So these structures are trimmed down really by this enzyme in a stepwise manner. And it sort of follows the paradigm of one glycosidic linkage related to one specific enzyme. But what's particularly interesting and that connects to the mission of GlycoNet in clinical research and biomedical research is the fact that the genes that encode this enzyme, and in particular mutation of these genes, give rise to a set of disorders known as lysosomal storage disorders. And for example, in the case of GLA, if you have mutants of the GLA gene that decreases the activity of alpha-galactosidase, leads to accumulation of the glycolipid substrate that can have a very detrimental effect for lysosomal biology in general, but also for human health. So this project is really started with a somewhat trivial question of how can we improve the way we study and we quantify this enzymatic activity, the turnover by glycosidases. And I'm fairly certain most of you will be familiar with how we do it routinely, which is the use of a substrate in vitro, either chromogenic or fluorogenic substrate that we put in presence of recombinant enzymes or even cell lysates. And we monitor changes in UV absorbance or changes in fluorescence. And this is really powerful techniques that are done routinely in our labs, and that are truly a cornerstone of biomedical research in our field. There's no doubt about it, and it will remain so for many years to come. But we start to realize that when you do that, when you break open a cell, you lose a lot of information, because it will be no surprise to you that these enzymes. I mean, I'm taking the example of glycosidases, but all of our target of interest could be used here. These enzymes, they operate not in vitro in a small microplate, right? They operate in a very crowded and busy environment, whether it be lysosomal organelles or other cell compartments. But anyway, in those environments, one could expect that you have numerous contact, protein-protein interactions or interaction with small molecules that are going to affect the activity of your enzyme of interest. And when you break open the cell, for example, when preparing a cell lysate, you inherently lose all that information. And so that's why the field of chemical biology has proposed to develop methods so that we can go and study that enzymatic turnover, that enzymatic activity within its physiologically relevant environment. And one approach to do so that has been very successful is the use of covalent activity-based probes. These probes are built around a recognition motif, in our case, most often a sugar. And you can have a tag that can be a fluorophore, but it can be really any tag. It can be a biotin for pull down. And the key feature of these probes is the so-called reactive warhead, because when such a probe binds to the enzyme active site, the warhead exploits the catalytic mechanism to create a covalent bond within the enzyme active site. And this permanently installs the fluorophore in your target. And because of that, you get a very, very sensitive and very powerful probes that can quantify a very small amount of endogenous proteins. One drawback, obviously, is that you do inactivate your enzyme active site, but nevertheless, this is an extremely powerful approach that can really give you good data on active enzyme levels. And there's a number of very successful studies in this field, several of them targeting proteases, notably by the group of Matt Boggio. And this is an example where you have a catepsin activity-based probe that has a fluorophore, a quencher, and you have that reactive warhead here. And this probe can label the cysteine within the enzyme active site and permanently install that fluorophore here. In our fields, there's also a lot of tremendous work done by, for example, Professor Overcleft, that are based around this motif of aziridine sugar analogs, and they can exploit the catalytic nucleophile within the glycosidase active site to create a covalent bond and install a fluorophore there. Another approach that I think is highly promising is the use of fluorescence quench substrate probe. And this one is in a way more similar to the way we do things in vitro, because you have an actual substrate of the enzyme. Very often you would want the fluorescence to be quenched so that you have your probe is dark. And when it binds to the enzyme and the enzyme does its job of cleaving, for example, a glycosidic bond, then you separate the fluorophore and the quencher, and you get a true readout of the enzymatic turnover by way of fluorescence. And we can see here that this has the advantage of not inactivating your enzyme. So in a way, you could get a really an actual readout of the turnover through signal amplification. But to be fair, it's clear that this approach has been less successful than activity-based probe, in part, I believe, because there are some very important challenges associated with the development of such probes. One of them, probably the biggest challenge, is the fact that because you don't attach your tag to the enzyme active site, your fluorescent product is very often free to diffuse away. And you very often have very big problem of signal stability. And because you have problem of signal stability, it's very difficult to get probes that give you quantitative data that really operate within a linear range within cells, and that give you good data to study the enzyme turnover. So that was sort of the state of the field and the challenges that we're facing. But when I joined David's lab, the lab had already some experience with the development of such substrates. My former colleague Anu Yadav and David Chen had worked on this type of molecules around the beta glucoside that had a quencher attached at the anomeric position and the fluorophore attached at the 6th position. And they showed that these can be processed by GCase, an enzyme that I'm sure you've heard about many times, and we'll discuss it a little bit further. But this probe can be hydrolyze the glycosidic bond, and you generate a fluorescent beta glucoside that can then be measured within live cells. And this was a tremendously successful approach. It was well supported by GlycoNet and enabled the screening of GCase activator, which is a very important problem for human health. And but the one problem when we started thinking about that is when we tried to apply the same sort of design to other glycosidases, it didn't turn to be broadly applicable to other enzymes. And it's not actually that surprising, because if you look at the state of the field, you realize that most of the successful approaches designing substrate probes have been targeting endo-acting enzymes, in particular endoproteases. And very often these endo-acting enzymes, they have that canyon-shaped active site topology. And if you realize that, you can see that you have your recognition motif in the middle of your probe, and you can visualize that it's fairly straightforward to attach a bulky fluorophore, a bulky quench on either end of that recognition motif. And then if the enzyme can cleave in the middle, you can separate the two, and you should be good to go in a way. In the case of exoacting enzyme and exoacting glycosidases in particular, the situation is often quite different, right? For example, in the case of human oglucnacase, you have a much narrower, a tight binding site that I believe it's quite clear that here, if you want to attach a bulky cyanine-based dye and a bulky quencher here on different position around your sugar, this is going to be a problem. So recognizing that, we started to think, how can we propose a better design to be broadly applicable targeting exoglycosidases? And we hypothesized that if we can connect the fluorophore and the quencher through a bisacetal motif. So this is the bisacetal motif is that sequence of oxygen, carbon, oxygen, carbon, oxygen. For the chemist in the audience, it's really hemiacetal anhydride, if you think about it. But we thought through that link, we could keep the fluorophore and quencher in very close proximity. So that should be good for efficient quenching. But also, obviously, it would completely preserve the carbohydrate recognition motif. The hypothesis is that if the glycosidase cleaves the glycosidic bond, you free up those two hemiacetals, and this one can actually spontaneously break down and separate the fluorophore and the quencher. One key point also is that this fluorophore would have an aldehyde functional group here that we thought could really help with the signal stability issue, potentially by forming transient imine bonds within acidic organelles of live cells. So we thought if we have a competent probe and good signal stability, we could get kinetics that are suitable for quantitative studies in live cells. So we first set out to work out a synthetic route, and it was largely inspired by the work of Professor Studley. And it started with the glycosylation of formic acid to give you these formyl glycosides. And these can be engaged in Wittig type chemistry to generate that vinyl glycoside that can also sometimes be referred as a vinylogose carbonate. And that double bond actually has quite a unique reactivity. because it can be engaged directly in a bromoalkoxylation reaction using N-bromosuccinimide with a spacer such as this one, and that generates directly that bisacetal motif. But interestingly, you can also generate the epoxide here that is quite remarkably stable. When I first isolated that, I was surprised that it was so easy to work with, and you can generate it using fairly mild condition using MCPBA. And that epoxide can actually also be reacted with the same spacer using acid catalysis to generate again a bisacetal motif. You may notice that the difference between these two is the quaternary center here that either bears a hydroxyl group or a bromine atom. And then from these intermediates, we did a three-step protecting group manipulation, the goal being just to free up that carboxylic acid here. And that gives us bifunctional bisacetal intermediates. And you could see here that we can pretty much attach whatever we want here through the carboxylic acid function group and the diazide here. We wanted to test the proof of concept in vitro, see, okay, can this design be turned over by enzyme? And we used the humanoglycnacase, which is obviously an enzyme that David Bocalo has a lot of interest in and a lot of experience. And when we use, we attach a simple fluorophore and quencher to these bifunctional intermediates. These are called E-dense fluorophore and dapsil quencher. They are very suitable for in vitro study, but not so much for live cells, but they're much easier to work with. So we started with these. And when we incubate these two probes with the humanoglycnacase, we could see very efficient turnover by the recombinant enzyme. There was linear with substrate probe concentration and with enzyme concentration. But what was also very interesting is that we did observe a very impressive quenching efficiency. And very importantly as well, these motifs, this bisacetal motif, proved to be stable across a fairly wide range of pH that was definitely compatible with the future use in live cells. So that was good proof of concept in vitro, but the key goal of this project was really to challenge this design and to test, can we apply it to different exoglycosidases? So going back to that scheme of lysosomal degradation of glycolipids, we turn our attention to GCAs, which is also a big interest in the Wakarchuk lab, and I'm sure many of you are familiar with the renewed interest in that enzyme, both because, of course, it's the primary driver of Gaucher's disease, but also because of the recent finding that a mutation in the GBA1 gene is one of the major risk factors for developing Parkinson's disease. So as you can see, this enzyme is the final step of the degradation of this glycolipid. And we set out to synthesize a glucose-based probe. This was done with collaboration with Evan in the Wakarchuk lab. And that's the sort of the beauty of that chemistry, is that you sort of just start with a different sugar and you just apply the same sort of chemical route, and you end up with an analogous probe. So the synthesis went okay, but when we tried to see if that probe was turned over by GCAs, we didn't observe any turnover whatsoever. So that was a bit of a setback in terms of versatility when your second attempt proves to fail. But we are not too discouraged because we observed that these were very nicely turned by OGA. We thought there must be something wrong here. And the only thing we could think of that could prevent the enzyme from doing its job is actually that quaternary center. We thought maybe the steric hindrance here can perturb the proper catalytic mechanism. So we thought, okay, maybe we just have to revise the design a little bit and come up with what we call the decongested structure that doesn't have that quaternary center. The small story here is that this quaternary center was part of the initial design because we thought this bisacetal might not be stable enough, but potentially through elimination, and we thought that quaternary center could bring stability. But when we observed that this was quite stable, we said, okay, let's give a shot to a simpler structure. Simpler structure doesn't mean simpler chemistry, but it's sort of similar. It's still based on B6 type chemistry and then one-step bromoalkoxylation. But then you have to add the extra step of a carbon-bromine reduction reaction that's done using radical chemistry. But this works, and we're able to synthesize the congested probe. And we are very pleased to see that once we synthesized this one, now it was beautifully turned over by the GCAs, and that really solved the main issue with the design. So that was reassuring, but still not a slam dunk in terms of versatility. So we sort of continued that to challenge the robustness of the design, and we turned our attention to alpha-galactosidase A, that's encoded by the GLA gene. That's the main that's the gene responsible for Fabry disease. And we thought it would be interesting also to challenge our chemistry, because now we would be exploring the chemistry in the alpha series, which shouldn't be too different, but still it's a way to see how far we can push the design. It's this alpha-galactosidase activity within lysome is interesting because there's actually two very closely related lysomal GH27, the AlphaGalA that cleaves so alpha-galactose residue, and the alpha-NAGAL here that cleaves N-acetyl gal residues, and they are very high homology. Aside from the fact that, as you can see here from the overlay of these two structures, so the blue one is the AlphaGalA and the yellow one is the NAGAL. You can see that the AlphaGalA cannot tolerate any modification at the two position. So, for example, it cannot act at all on an acetylated sugar here. As you can see, this the co-crystal with the DJJ NAC, which is a specific inhibitor of NAGAL that has that NAC residue, clashes with that residue on the GALA enzyme, the blue surface and residue here. What we have here also in blue is the migalastat compound, which is a pharmacological chaperone, which is actually approved and used for treating Fabry disease. So here the migalastat is crystallized with alpha-GalA and the DJJ NAC with the NAGAL. But we saw that as an opportunity to test our design and see, okay, can we generate probes for these two enzymes? So I won't go into all the details of the chemistry again, because it's fairly similar. It starts again with the B6 type chemistry and then activation of the double bond. But thanks to the hard work of my colleague, Dr. Roger Ashmus, we're able to generate both the alpha-gal probe and the alpha-gal-NAC probe. Once again, with a very simple fluorophore and dapsil for just an in vitro test. And when we tested first the alpha-gal-lactose probe, we saw that this probe was turned over by both alpha-GalA and alpha-NAGAL, whereas the alpha-gal-NAC probe was only turned over by the NAGAL enzyme, not at all by the GalA enzyme. So that was consistent with what we inferred from the crystal structures. But we thought it was really time to go in live cells and test the design, test the performance of the design in live cells. So you can't really use Eden's dapsil fluorophore and quench pair in live cells because they're not really performing well enough. But thanks to the bifunctional design that I showcased earlier, the fact that you have a carboxylic acid here and azide here, you can actually attach commercially available fluorophore and quencher or ones that you synthesize yourself. But these molecules are big and quite complex, and it's quite nice to be able to just, at the end of your synthesis, attach these molecules. So we did that. We connected the TAMA-based fluorophore on both the alpha-gal scaffold and the alpha-gal-NAC scaffold. We checked in vitro using recombinant enzyme, and as we observed for the previous probe, for the alpha-gal probe with the TAMA residue, it was turned over by both enzymes, but much faster by the GalA enzyme. Expectedly, once again, the alpha-gal-NAC probe was only turned over by the NAGAL enzyme. So what we had before going into live cell, we thought was a very fast and selective alpha-NAGAL probe that should be able to only report on the NAGAL turnover in live cell if it works. But we're not too worried about the alpha-GalA because we thought we could actually isolate the turnover AlphaGalA, isolate the signal coming from the GalA enzyme using the NAGAL specific inhibitor that I presented earlier, the DJJ-NAG. We thought if we can inhibit that NAGAL enzyme within cells, then we can isolate and measure specifically the GalA turnover. But we were very eager, obviously, to test them in live cells. And we started using human SK and SH cells. We started with simple inhibition control. So the way this is done is just you take live cells and you treat them with your probe, you incubate for a couple of hours, and with or without inhibitors, and then under a microscope, you acquire the fluorescent signal and integrate it and relate that to a percentage of activity. And what we observed with the AlphaGal probe is that treatment with Migalasta almost completely abolished any fluorescence. A mixture of Migalasta and DJJ-NAG did the same, but DJJ-NAG, the NAGAL specific inhibitor, didn't seem to have much of an effect. If you treat the same cells, but with the AlphaGal-NAG probe, now you get a different profile altogether, because the DJJ-NAG is now, as expected, a very potent inhibitor and completely kills your fluorescence. Whereas the Migalasta, which still binds to the NAGAL, but is a sort of moderate inhibitor, only partially reduces the signal. So this was indicating that what we are seeing is here specific signal rising from the turnover of the probe by AlphaGalA, and in that case, specific probe for NAGAL. So that was very encouraging. But what was truly groundbreaking for us and for this project is when we started to do dose and time response of this probe, trying to optimize, find the perfect condition to operate within live cells and to get a linear range, linear response. And what we observed is that after a couple of the first few hours of incubation, we see a little bit of a lag phase, presumably due to the uptake of the probe and establishment of steady state. But after that, we did seem to reach a linear range, both in terms of substrate concentration, but also with respect to incubation time. And I think this is possible thanks to the next piece of data, which is the signal stability in cells. And it doesn't look like much, but this is probably the most impressive result, and or at least one of the most satisfying one, because what we did here is we treated the cells with the glycosidase probe for a couple of hours, 2 hours, and then you wash away your cells. You wash all the probe away, and you do a couple of washes, and then you add imaging media that is supplemented with a high concentration of inhibitor. The goal is to kill any sort of enzymatic turnover once you've added the imaging media. And what you do then, so the T0 is your first imaging of those cells. And what you do is you keep imaging the same cells over and over again for a period of 3 hours. The goal is to see, okay, the fluorescent signal that was generated within the first 2 hours, how stable it is within the cells. And this, we found that this was quite remarkably stable. And we definitely think that we're able to see a good quantity of data in live cells, in large part thanks to that good signal stability. So because we're able to operate within that range, we thought we could then take it to the next level and do measure target engagement within live cells. And this is quite unique in that these are image-based IC50 measurements in live cells. So it's based on integration of the fluorescent signal. And we can see that by using various concentration of Migalasta and our AlphaGal probe, we see that nice IC50. Interestingly, the DJJ-NAG, various concentration of it, doesn't seem to have any effect on the signal using the AlphaGal probe, indicating that this probe seems to be at least functionally selective for the AlphaGalA in that cell line. Importantly, if you use the other probe, the AlphaGal-NAG now, what you see is that the DJJ-NAG now is a very potent inhibitor as expected, and you can measure that IC50, whereas the Migalasta is order of magnitude less potent in live cells. If you can do that, we can also work on clinical samples and quantify the activity, for example, in Fabry patient fibroblasts. So these are a series of cell lines, fibroblasts from patients suffering from Fabry disease, and that have different mutations. that either completely kill the enzyme, completely reduce the presence of the enzyme, so where we'd expect no activity whatsoever, and some mutants that actually should show reduced activity. And when we treat these patient fibroblasts with our probe, what we saw is indeed no turnover whatsoever in those two cell lines, and reduced activity in R301J below 20%, and that's consistent with what's known of this cell line in the literature. What's interesting as well, an important control, is that if you treat those cell lines with the AlphaGalA probe, these cells don't have any mutation on the NAGAL gene, right? So you'd expect something that's not really affected, and indeed we didn't see much of an effect of the different cell lines on the total fluorescence. But importantly, that fluorescence was completely abolished by the DJJ-NAG, so the NAGAL specific inhibitor. Then we also wanted to test whether we could use these probes to study small molecules, so small perturbators of lysomal biology, and compare that with sort of the standard approach, right? What you would do using lysates. So you can take your cells, treat them with small molecules such as monantine, brepheldin, and bifidobacillin that either impact trafficking, cellular trafficking to the lysome, or that perturb the endocytosis and lysomal fusion. And if you do that after you've treated yourself with these compounds, you can incubate them with our probe and measure the fluorescence in live cells. And what we observe is that these treatments seem to completely reduce drastically, at least, the turnover within lysome. But if you break open these cells and study the lysate assay using 4-methylamberifol alpha-galactoside, then you see a completely different activity profile, which presumably is due to the fact that if you impair the trafficking, you don't necessarily disrupt the synthesis of your protein, of your enzyme target, right? So if you break open the cell, you might measure activity coming from mislocalized protein as well. So this, I think, really showcases the value of going directly into live cell, measuring the relevant activity in its relevant environment. One feature that I really like to show, because I think it's a really unique thing that you could do with substrate probe when the fluorescence quenching gets good enough, is that you can take live cells that are inside the microscope in their imaging, in the actually growth media. I think this one has phenol red, FPS, everything, and you can just spike your substrate probe in it and start recording fluorescence. And what you see over the course of 2-3 hours is the apparition of that punctated signal, that fluorescent signal, consistent with the lysomal turnover of your substrate. And I think this is something really unique that you could only do with a fluorescence quench substrate probe approach. And maybe I'll finish on that story by reminding you how that GlycoNet had a very important role in that it supported the Center for High Throughput Chemical Biology at SFU, that enabled us to do a lot of this work and a lot of the optimization in a miniaturized way. A lot of these dose response, time response were done in full 384 well microplates, and that also shows that these type of approach are amenable to high throughput screening. And maybe that's one thing that I would like also to highlight here, is the value of these live cells measurement, right? When you have a target, an enzyme that you're trying to develop better small molecules to potentially increase its activity, for health benefits, very often, if you go with in vitro test and you screen compounds, you may find direct activators, small molecules that act directly on your protein, your recombinant protein, for example, or through, allosteric mechanisms. But what you're missing is all the potential impacts on your activity that are indirect, right? If you do screen directly in live cell, you may identify compounds that act indirectly on lysomal biology to increase your target activity, or it could act at a different level, a transcriptional level. what the key thing is that you open up a lot of other possibility and a lot of possibility for discovery of interesting molecules. So I think these two approaches are definitely going to be complementary, but there's a lot of potential value in live cell phenotypic screens, such as the ones that are enabled by these chemical biology approaches. I'd like to finish by saying that here we are continuing to work on trying to get better tools for monitoring glycosidase enzymes, and it's sort of the same thing that I was mentioning at the beginning. What's sort of the end goal, the end goal for getting a good substrate probe. If we think about how we're going to quantify enzymatic activity in, let's say, 10, 30 years, I think we're going to see more and more of these precision tools and probably less and less, Western blots and in vitro tests. But one thing that I keep coming back to is when we design these, we tend to assume that if you want a good substrate, you want a fast substrate. We assume the faster the better, and I think maybe it's time to revise that, or at least test that idea. There might be some optimal kinetics that are really important for quantitative chemical biology. And the important thing here is that if you want to fine-tune your chemical probe's kinetics, you need good chemistry. You need a way to generate chemical diversity around your design, around your scaffold, and that's not something that was really possible with what I just presented. Indeed, the chemistry works well for the bisacetal, but it would not be easy to generate different motifs with electron withdrawing group, with aromatics, with phenols. That would not be easy. So we actually start thinking about it, and we thought, okay, if we want to just bring diversity there, can't we just directly glycosylate transiently form hemiacetal in sort of a one-part, three-component glycosylation? So we're starting to work on that, and we have very exciting preliminary results that you can actually mix an aldehyde and, for example, a phenol. You're going to generate some hemiacetal, and if you can trap that hemiacetal with a activated glycosyl donor, or even better with a free acid, you could generate bisacetal motif. So this is all preliminary. Hopefully, I'll be able to present you more definitive results. But we do see that we can generate these structures this way. The yields are still quite low. I mean, two digit low, but it's not that bad. But there's a lot of work still to be done here. Another approach that we're working on in the lab, we are also trying to apply the same philosophy in the field of glycan-protein interactions. And we're working in synthetic biology approach and protein engineering, but also trying to develop chemical probes. With that, I'd like to thank, obviously, David Boccallo, my former supervisor, and my former colleagues at SFU from the substrate probe subgroup, and also the students here in Montreal that have joined the lab. And I'll also want to thank particularly GlycoNet for the support and the current funding that I'm receiving now at University of Montreal. Thank you all for your attention, and I'll gladly answer some questions. Thank you.